Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about photosynthesis. I love photosynthesis because it gives me two things that I need. I need to breathe, so it gives me oxygen, and I need to eat, so it's going to give me food, and so I love photosynthesis. You might think it's only found in these things, plants, but it's also found in bacteria, and it's found in algae, and so it's found in protists, and it's found everywhere. And so photosynthesis has been around a long time. It's super important that you understand how it works. And so let's start with the site in eukaryotic cells of photosynthesis, and that's the chloroplast. So this is a number of cells, and you can see how many chloroplasts we could have in a typical cell. So there's a whole thing. There are a few terms you should be familiar with and where they are. First one is thylakoid membranes. Thylakoid membranes is going to be organized like this, and basically that's where the light reaction is going to take place. If you got a stack of thylakoids like this together, we call that a granum. The other big thing to understand photosynthesis is that it is filled with a liquid, and that liquid is called the stroma, and that's going to be the site of the Calvin cycle. If we were to grind up a leaf, that we could find is that there not only one pigment, Rho5A, that does photosynthesis, but there are a number of them that are working together. And so if you grind up a leaf into some chromatography paper and then you put it in a solvent, what you get is chromatography. It's going to separate into all of its different parts, and so this right here would be chlorophyll and A and chlorophyll B, and this would be like carotene and xanthophylls, and they are all working together. If you see this other pigment in the fold when the cyclophy moves back into the leaf and is reabsorbed. But if we look at what light they absorb, here's a chlorophyll A and here's B. This is what called the absorption spectrum, and what color of light they are able to absorb, and you can see that they absorb a lot of the blue, a lot of the red, but they don't absorb a lot of this in the middle, this green. And so a quick question could be what is their at least favorite color plants? And the right answer would be green, because it reflects that green light. Now this is actually puzzle scientists for a long time, and we really don't have a definitive answer as to why plants are green. Know that if they were black, they probably would get a little bit too hot. They would absorb too much light. And let's start with an equation, because this is simple reaction. It is chemical reaction with a number of steps. But what are the reactions? Water and carbon dioxide. And so how does a plant grow? It's basically taking water in from its roots and is taking carbon dioxide in through its leaves, through its stomata. The other thing is needs is light, and so it's just taking these simple ingredients and then it's weaving those together into glucose, these monster molecules here, and then oxygen. And so this is the food that I get, and this is the oxygen that I breathe now. Are plants just nice? No, they are making this sugar for themselves, though, so they can break it down during cellular respiration. And in fact, if I put this around in the other direction, that becomes cellular respiration. So they are making food for themselves, and they are also going to make some of the structure. So like the cellulose in the cell wall of a plant is made from that as well. Okay, so whenever I try to think that what are the different steps in photosynthesis, I always imagine this picture right here. There's a photo and synthesis in the world. Photo means light and synthesis means to make, and so there are two steps. In photosynthesis, the light reaction, and those are going to take place in the thylakoid membrane, and then the Calvin cycle we used to call this the dark reactions, which is a silly term. Doesn't happen during the dark, and it happens during the light, and so basically the person who worked this all out is Melvin Calvin, and so we named it after him. Where does this take place? You guessed it. It takes place in the stroma, or this liquid portion. And so let's kind of to do a cartoon version of photosynthesis. What are the reactions? Again, water, light, and carbon dioxide. What are going to be the products that come out of this? It's going to be oxygen and glucose. So let's watch what happened. It depends reactions. Water and light go into the thylakoid membranes, and they produce two dependent reactions. They produce oxygen. Oxygen is simply a waste product, and then they are going to produce these chemicals, the NADPH and ATP. So they have energy now. Let's watch what happens to them. Well, the energy is going to transfer to Calvin circle while carbon dioxide comes in, and then glucose goes out. And so this is big picture of photosynthesis. But now let's kind of dig in a little bit deep and talk about the light reaction. Okay, so where are we? We are in thylakoid membrane. So we are in this membrane right here. So if we will go to zoom into that membrane right here, that's what this diagram is. Okay, so what are the two things coming in? Well, the first one is going to be light. So light coming here in here, light coming in here. What's the next thing that we are going to have coming in? And that's going to be water. Okay, so let's look at some of the other big features in this thylakoid membranes. So this is the outside, or the stroma, and this is going to be the lumen, or the inside. And so there's a couple of big things right here. What's in here? Well, there's basically going to be proteins with chlorophylls on the other side of it, and so we call that whole thing together a photosystem. So this first one is actually called photosystem 2, and then we go to photosystem 1. And the reason we go backward is that photosystem I was discovered first. So basically what comes in? Light. What's light used to do? Well, that light is used to power the movement of an electron through an electron transport chain. So that electron is going through proteins, carrier proteins, and eventually that electron is going to go here. Is going to, because it, remember that one of the products of light dependent reaction. Okay, what happens to the water then? So the water is going to be split right away. If you split water, what do you get? Well, you get oxygen. So that's the O2 that going to diffuse out of a cell, and that's the oxygen that you are actually breathing right now. And then we are going to have these protons, which are simply hydrogen ions. So they are hydrogen atoms what have lost their electron. Okay, so this is getting kind of messy. Happen next, and as that electron move through the electron transport chain, and again is powered by the introduction of light here and light here, that electron is going to be moving all the way down here, and every time it goes through one of these proteins, it's pumping protons to the inside. So it's pumping protons to the inside. Now protons have a positive change. So basically what's happening is that you are building up a positive charge on the inside. So there are a positive charge in here. If you know how cellulose respiration works, you will realize that this is the opposite of that. So now we have all of these positive charges on the inside. Where do they go? Well, there are only one hole that they can go through, and that is to go through this protein here. And those protons move out. They're moving through a protein called ATP synthase, and it works almost like a little rotor, and every time a proton goes through, we make another ATP. So what have we made in the light dependent reaction? We have made NADPH, and we've made ATP. And what's nice about that is they are now just sitting right here in the stroma, and they are able to go on to the Kelvin circle, which is going to be the next step in this process. And so who's providing the energy? Light. Who's providing the electrons? Water. And then a waste product of that is simply going to be oxygen. Okay, let's go to the Kelvin cycle then. So what's happening in the Kelvin cycle? You can see here those reactants. So we've got our ATP here, and and ADPH. What are they providing? Simply energy. You also have this molecule here. It's called RUPP. Basically, it's a 5 carbon molecule, and then we have carbon dioxide coming in. So it moves through the stomata of the leaf and is going to diffuse away in. Carbon dioxide is a 1 carbon molecule. So basically there's an enzyme here called riboscope, and it's going to attack this 1 carbon molecule to a 5 carbon molecule. It immediately breaks into 3 carbon molecules, and then it gets energy from ATP and NADPH. And when we are done, it is creating this chemical down here called G3P. What does G3P become? Well, it can be assembled quickly into glucose or sucrose or maltose or whatever they need to do. That's going to be produced right here by the G3P. So what we are synthesizing, where we are synthesizing. In other words, we are taking carbon and we are fixing it. We are making it unusable. That G3P is released, but a lot of it is recycled again to make more of this IU. And so that's why a cycle over and over again. What's the big picture? If we don't have ATP, if we don't have NADPH, then this process is going to shut down. What's the other thing that could shut it down if we don't have carbon dioxide? Okay, so that's basically photosynthesis, and again it's being worked for billions of years, but there's a slight problem, and that problem is called photorespiration. What's photorespiration? Well, photorespiration occurs only when we don't have enough carbon dioxide. So if we don't have enough carbon dioxide, let me cross that down. Well, we certainly can't make our G3P, but something worse happens. Oxygen can actually jump into the carbon cycle, and during riboscope can form another chemical. Now that chemical doesn't do anything. In other words, it has no purpose, and the cells actually has to break it down. And so as a result of that, plants, and we call almost all plants C3 plants, and the reason we call them C3 plants is this. G3P is going to be a 3 carbon molecule. So for these C3 plants, photorespiration is bad. In other words, they don't get anything out of it, and so they are going to lose based on that oxygen kind of jumping into the carbon cycle. So you might think evolutionary. Why would this have even evolved? Well, remember, photosynthesis shows up first, and then oxygen in the atmosphere shows up much later, and so it wasn't a problem initially, but it became a problem. Another question you might think is, when are we not going to have enough carbon dioxide? When wouldn't we have carbon dioxide? Well, how do they get carbon dioxide? And plant is going to have a stomata and is surrounded by guard cells. And so basically when a plant opens up its stomata, carbon dioxide can diffuse in, and so the only time the plant wouldn't have carbon dioxide because we have tons of carbon dioxide in the atmosphere, where it is when it's actually closed, and when it could be closed in a plant. The only time it's closed is when it's really hot and a plant doesn't want to lose water, because through transpiration you are constantly losing water. And so if you are a plant, if it's a hot day, you have this really tough choice. If you open up the stomata, you're going to lose water. You could saliva up. If you close it, you can't get carbon dioxide. You're going to start doing photorespiration, and so of course Nitro has come up with solutions to be this over time, and it's only going to be found in plants that live in really hot environment. So here's the first solution, and this totally makes sense. This is in CAM plants. CAM plants could be a jar plant or like a pineapple. Basically what they do is they only open their stomata at night, and so at night they open their stomata, and then the carbon dioxide will come in and they will create malic acid out of it. So they are going to store it in vacuoles inside the cell. Okay, so now when it's daytime, what they can do is they can close the stomata because they don't want to lose water. And now we actually take that carbon dioxide out of the malic acid, and they can use it in the Calvin cycle to make sugars. So the great thing about CAM plants is, again, they are only taking in carbon dioxide at night when it's cold, and then during the day they can close their stomata and they don't lose water. Another example of this would be the C4 plants. What they do instead of doing it day and night, what they do is they take that carbon dioxide in, and they actually use enzymes to make a 4 carbon molecule out of it. That 4 carbon molecule will move to some cells on the inside of leaf called the bundle sheath cells, and then they can simply introduce carbon dioxide into the Calvin cycle here. And so again, both of these solutions are basically taking in carbon dioxide when you can get it, creating a chemical out of it, and then they can introduce that chemical into the Calvin cycle, and they don't have to wait for carbon dioxide to refuse in. Now, of course, there are going to be extra steps in here, so it's going to require more energy, and so we only see this in areas where it's really warm. But an example of a C4 plants that we are eat and use a lot. In fact, most of us are used made out of this stuff. is coined, and so that's photosynthesis. The simple problem is photorespiration, but I hope that helpful. Thanks for listening.